Hello, I am going to start plant breeding for disease resistance. Resistance is defined as the genetic ability of a plant to prevent pathogen from causing diseases. These are the definitions of resistance. Resistance is defined as the genetic ability of a plant to prevent pathogens from causing diseases. There are some advantages of plant breeding for disease resistance. The development of resistance in crop enhances productions. These are the first advancement. That means कि enhances the productions of crops. Second, reduces the dependence on fungicides and bactericides. These are the major advantages of resistance. Now, there are some diseases caused by the pathogens like fungi, bacteria, or viruses. So, disease can be caused by fungi, bacteria, or viruses. You see the chart, there are some diseases here caused by fungi, bacteria, and virus. Causative agents like fungi, which is cause the disease, like brown rust of wheat, red rot of sugarcane, lead blight of potatoes. These are the disease which is caused by fungi. Black rot of crucifers, blight of rice, and citrus cankers are the diseases which is caused by pathogens like bacteria. Viruses, also called tobacco mosaic and turnip mosaic, are the major disease caused by the fungi. So, these are the some diseases which is caused by fungi, bacteria, or viruses. Now we discuss methods of breeding for disease resistance. There are two methods for carrying out breeding. First is conventional breeding method, and second is also known as mutation method. First is conventional method and second is mutation breeding. Mutation breeding. So now we discuss conventional breeding methods. Conventional plant breeding has been going on for hundreds of years and it is still commonly used today. All our major food crops of today are derived from domesticated varieties. It involves hybridization of pure lines and selections of plants with desirable qualities and is carried out by the following steps. So there are the various steps involved in the conventional breeding methods. First, selections and screening. Selection and screening of germplasm for disease resistance. Second, hybridization of selected plants. Hybridizations in which two desirable characteristics crossed with each other to produce the desired characters, hybrids. Selections and evaluations of hybrid. And last, testing and release of new varieties into the market. Here, the sum of varieties developed by conventional breeding methods are crop like wheat. Its best variety is Himgiri, and disease resistance to diseases leaf and stripe rust and heel burnt are the disease which is resist to the variety of Himgiri crop like wheats. So crop like wheats, its varieties Himgiri, and it is also resistant to disease like leaf and stripe rust as well as heel burnt. Second, crops like brassica. Its variety is Pusa swarnim and disease resistance is white rust. Second, like brassica, like mustard. Karan rye, that is also resistance to disease is white rust. Crop like cauliflower. Its best variety is Pusa suvra, Pusa snowball K1. It is resistant to disease red rot, curl blight, and black rot of plant. Next, cowpea. Its best variety is Pusa komal, and disease to resistant is bacterial blight are the resistance to disease. And then chili. Next crop is chili. Its best variety is Pusa sadabahar, and disease resistance is chili mosaic virus, tobacco mosaic virus, and leaf curl are the disease which is caused by viral infection. So now these are the so many disadvantages of this method, like limited number of disease, resistant genes are present. These are the demerits of these methods, conventional methods. And second, limited number of disease resistance gene have been identified in crop varieties or wild relative. So these are the disadvantages of these methods in which limited number of disease resistance genes are also present. So these are the conventional methods of breeding of plants. The second method is known as the mutation breeding. First, we see the mutation. Mutation is a phenomenon by which genetic variations is achieved through changes in the base sequences within genes which create a new characters or trait absent in potential generation. So, mutation breeding can be defined as the improved of crop by changing the genotype of plants through induced mutation. It is through induced mutation. It is included artificially through use of chemical or radiations like gamma radiations. It is carried out by the following main steps. So there are the different steps followed in the mutations breeding. Mutation breeding is defined as the process of breeding by artificially inducing mutations using chemicals like aniline or radiations like gamma radiation. So these are the different steps including mutation breeding. First, inducing mutation in plants by various means. Second, a screening of the plants for resistance. And third, selecting the desirable plants for multiplications and breeding. These are the different steps by which mutations breeding is found in plants. In mung bean, resistance to yellow mosaic virus and powdery mildew were introduced by mutations. In Vindi, resistance to yellow mosaic virus was introduced from a wild species from a wild species resulting a new resulting a new varieties called Parbhani Kranti. Parbhani Kranti. So these are the mutations breeding in plants. So plant breeding methods for resistance to disease are of generally two types. One is conventional method and second is mutations breeding methods. So conventional breeding methods in which germplasm for disease resistance and hybridizations of selected plants, selections and evolutions of hybridization, testings and release of new varieties into the markets. These are the methods, and in mutation varieties it is affected by the gamma radiations as well as the chemicals like aniline. So these are the steps inducing mutations in plants, screening the plants for resistance and selecting and desirable plants for multiplications and breeding. So these are the steps, various steps which is also included in the plant breeding for disease resistance. So now we discuss the pest resistance variety of the plants. Plant breeding for developing plant breeding for development resistance to insect pest. Insect pest. So these are the another methods for development resistance to insect or pest against. The host plants may be resistant to insect pest due to morphological, biochemical or physiological characteristics. So it is affected by insect pest due to affected by morphological, biochemical, and physiological characteristics. Of plants. So these are the some characteristics that lead to pest resistance are characteristics that lead pest insect resistance are hairy leaves in plants, for example, resistance to Jaceids, resistance to Jaceids in cotton and cereal leaf beetle in wheat. Second characteristics, solid stem in wheat. Stem in wheat exhibit non-preference by a stem exhibits non-preference by a stem sawfly. Third characteristics, in cotton, a smooth leaf and absence of nectar repel bollworms. Fourth, in maize, high aspartic acid low nitrogen and sugar content protect them from stem borers. So the steps of breeding method is same as for the other agronomic traits, and some variety developed by the hybridizations and selections of the different plants. Some varieties develops by hybridization. Varieties developed by hybridizations and selections are Some varieties developed by hybridization and selections are crop like brassica, that means repi, seeds, mustards, seeds, mustards, rapeseed mustard. Varieties Pusa Gaurav, an insect pest like aphids. Crops like flat bean, varieties Pusa Same 2 and Pusa Same 3. Pusa Same 2 and Pusa Same 3. Pusa Same 3. Insects like jassids, aphids, and fruit borer. Crop like okra, bhindi. Okra, bhindi. Varieties like Pusa Swarni, Pusa A4, Pusa A4. Savani, insect like shoot and fruit borer. So these are the different crops varieties developed by hybridization and selections, its insect pest and varieties. Thank you.